Su trabajo incluye el análisis farmacológico de especies de plantas, insectos y hongos que nunca se han estudiado en el laboratorio. Fabien realiza actualmente investigaciones farmacológicas en 5 países y posee una amplia experiencia en el estudio de la aplicación de remedios naturales en la medicina tradicional en comunidades indígenas. Y también con nosotros, y Manuel y yo, somos los EC50 en 1922.
Thanks for the kind introduction, Alicia. I didn't understand one word, but I think it was okay, positive, yeah, not negative. Okay, so yeah, I'm really happy to be here today in Spain, in Ciudad Real. Thanks for the invitation, Manuel, back there, yeah. So my name, as Alicia, is Fabien Schultz, and I'm leading the Ethnopharmacology and Zoopharmacognosy Research Group in Hamburg, Germany, at the Bernhard Nocht Institute for Tropical Medicine, likely better known as the Tropical Institute of Hamburg, at least in Germany it's known for that. And today I'm going to talk about, from forest remedies to lab discoveries, how mountain gorillas, chimpanzees and elephants inspire the future of medicine. So ethnopharmacology and zoopharmacognosy, likely these are not terms that you're familiar with. It's niche fields in science. And I'd like to start with giving you a definition of this, or at least of my interpretation of these fields. Ethnopharmacology is the science of ethnopharmacology seeks to investigate humankind's use of natural materials for medicinal purposes. So these natural materials may be plants, most known like medicinal plants, but they can also be insects, mushrooms, minerals, microorganisms, or even in some cases animal secretions. It is a highly interdisciplinary field, often also described as a bridge between the medical or natural sciences and the humanities or socio-cultural sciences, because there's many or various backgrounds and methods applied from diverse fields. In my interpretation of ethnopharmacology, we usually initially document traditional uses, and then we also do pharmacological testings at the lab. So I'm lucky because I'm working in the field as a field researcher. I'm also an office person, but I'm also leading 2 laboratories in Germany where we do real lab and drug discovery work. And we also, or my wife and I, we have an NGO where we do community projects also as part of the research. And if this sparks your interest, we've published twice about this, targeting newcomers or basically non-scientific people or non-ethnopharmacologists that are interested in this type of work that we do. Let's talk about zoopharmacognosy. Is there anybody who knows what that term means or has a guess? Huh? So zoopharmacognosy is the science of animal safe medication and the pharmacological investigation of the drugs. It's part of a broader field of One Health, basically a hot topic in science right now. And the methods are quite similar to ethnopharmacology. It's just a field research which is different because we are monitoring animal behavior and not documenting knowledge and interviewing human indigenous communities, or humans in general. But then the lab work is quite similar. So today I would like to give you some insights into the world of an ethnopharmacologist and a zoopharmacognosist, and I try to show a lot of pictures and show a lot of videos, less lab data, but really guide you through all the aspects of the work we do. And the Swiss alchemist Paracelsus, the father of the medical revolution and the Renaissance, he once said, "All that man needs for health and healing has been provided in nature. The challenge of science is to find it." And it's also like a little bit of spirit of an explorer, that you're like going somewhere where nobody has ever gone and trying to find something, to gain more knowledge about something. And I also find this sentence still very actual for my research mission. Here you can see some of the fieldwork locations where we currently have projects and where we are working. So most of them are in Sub-Saharan Africa. Today I will only provide some insights or some of results on my work in Uganda. Right now there are at least 39,000 plant species that are used medicinally worldwide. However, very few of these have ever been taken to a laboratory and looked at under the lens of modern science. So Gonzalo said, so this is just plants, but Gonzalo earlier said that there's only around 10% of the world's insect species being described by science or discovered, let's say it like this, because there's indigenous people or local people that have discovered it likely. But imagine how many of these have been taken to a lab and looked at for the secondary metabolite profile, so for their potential active ingredient, the chemical profile. It's such a low number. And it's really alarming if you think like of the high demand for more and effective and new drugs now and also in the future. Let's talk about antibiotics and the antibiotic resistance crisis, one of the major threats to global health worldwide. The O'Neill report commissioned by the UK government recently said that by 2050 there will be more people dying worldwide because of antibiotic-resistant bacteria than currently people are dying because of cancer. So this is really alarming. But at the same time, as an explorer, it's also very exciting because there's so much to discover, so much room for to unlock nature's secrets. And you may be wondering, why is this even relevant? We have like in the Western world, we have our modern pharmacies, we have the drugs in there, but very few people know that there's also great precedence of natural products in modern medicine. Around 50% of all drugs, authorized drugs in our pharmacies right now, they are derived either directly or indirectly from natural products. So it's really relevant. When working in traditional medicine in general, or in our zoopharmacognosy studies, we actually work with and we observe wild animals. And right now, these are mountain gorillas, chimpanzees, and elephants in 3 countries. Yeah, I'd like to tell you a story now. So this is Tom. Tom is a male chimpanzee from western Uganda, and up until his death, he was a member of a community that is part of our studies. One day, Tom got caught in a man trap laid out by the local people that found themselves competing for food with the chimpanzees. He died a couple of days later due to the infected wound, and he just saw that he was pointing at his infected wound, and there's a close-up picture coming up in a bit. However, just after this injury, Tom excessively began eating leaves of a local plant called mbiribiri, a plant that he or none of his community or family members has ever been seen eaten before, according to the local primatologists. And here you can see a close-up picture of the wound. So why is Tom doing this, or why did he do this? Was he eating this specific plant in a desperate attempt to save his life? There are still so many things we don't know about mountain gorillas, about chimpanzees, elephants as well in my case. And one of these things is if and how they self-medicate. Behaviors like Tom's, not only in life-threatening situations, suggest that they actually do. So what if we as humans can learn from Tom and his fellow apes? What if human lives can one day be saved by following our ancestors' lead? So far in this particular project, we've worked at 4 study sites that were quite different. There's the moist evergreen rainforest in Chibale. There's the moist semi-evergreen Budongo rainforest, where we just published a big paper last year, and the dense mountain rainforest in Burundi. And then there's the highly disturbed farm forest habitat in Bulindi, which is obviously where human-wildlife conflict is quite big, and Tom used to live there. And this fieldwork was also assigned an Explorers Club flag expedition in 2019 and then in 2022 again, where we led a team of researchers to these 5 study sites. And one of the main objectives was really for the first time to document the safe medication behavior in mountain gorillas and in that specific sites and in chimpanzees as well. And then also in many cases for the first time to take samples to the laboratory. And I'm going to talk about the laboratory results as well. Traditional healers in these indigenous communities that live nearby, they were also interviewed, and this is also always really important in my research because we look at human overlap and human uses as well, so it's all interconnected in general. And in all my projects, that's why I'm saying I rely on the help of local traditional healers or local people in general. Their knowledge on natural remedies goes back numerous generations and is only transferred orally from generation to generation. And then when selecting species, putatively medicinal species for the research or for the lab work, we also rely on these local traditional healers or local people that live there, because in many cases it's said that their knowledge is already lost or almost lost. Worldwide, traditional knowledge in indigenous communities is generally already been lost, and so this first-time documentation of knowledge or of traditional knowledge is really essential for future generation in the light of the demand for the search for new drugs in the future. Therefore, so the loss of culturally and scientifically valuable information is a global problem that's actually affecting all of us and not just the people that live in these regions. Hippocrates once said, "Let food be thy medicine and medicine be thy food." So how can we distinguish, let's switch back to the animals, how can we distinguish between normal feeding behavior and actual safe medication behavior, therapeutic safe medication behavior? We can make direct observations, for instance, so such as Tom's injury, like a wound, and there's been a couple of papers out where there was an orangutan who put a leaf of a plant on a wound. And running noses, diarrhea, all these things, and unusual feeding events, these are usually the clues. For instance, this chimpanzee, he traveled 20 kilometers away from his community to a swamp area and to access this plant that only grows there. And also the way he's eating it, you see he's discarding the leaves and the stems and only eating the petioles, which is quite unusual. And when we collect this species, we do it the same way. So we also separate the samples into, we also separate, thank you. We separate the samples into different parts and to look at the chemistry, whether there's different active ingredient composition in different parts of the samples. And then there's another method to look at indirect, or to do indirect observation, for instance. And this particular behavior here, we look at, for instance, at whole leaf swallowing, which is an established behavior in chimpanzees, actually. So these leaves on the right side, they are found in the feces, along with parasites. In this case, it's Oligophagestonum load that was reduced. And it's also in these cases that, it's a pathogenic nematode, that there was a lower load. So this is Anelima niacense, which is a plant that we study. And then, we collect all these species. There's Henrique and Candice, who are two of my PhD students who've just been in the field, just returned after 2 months in Uganda, and they collected lots of species and take them to the lab. But before talking about lab results, I'd like to talk a little bit about further steps, what we do in the field, or when we come back. So we interview local traditional healers for the overlap as well, but something we also do, which is basically complying with the Nagoya protocol and to create access and benefit sharing benefits, non-financial or financial benefits for local people. So all the studies we do, we transfer back the results into workshops with the local people, and we also initiate community projects. So there's been one of the examples, the Jiggers Medical Intervention. Here you can see Charity, who, like 1 year after I worked with her, this is a Batwa community, by the way, so it's in Burundi, it's former hunters and gatherers that are severely discriminated politically and socially in the Ugandan country of Uganda, I mean. And they were not allowed to access the forest anymore to get the medicinal plants for treatment when the COVID pandemic hit. So many children, she called me and said, our children are dying because of a disease called jiggers. And we found that this is basically bed bugs, it's a parasite disease, but due to secondary infections, there's basically a sepsis and the children die. And what we did, for instance, is we brought in medical professionals. Pointers? Yeah, no. And these medical professionals, they treated in a week 120 children. They trained from 17 Batwa communities. They trained the parents sustainably how to remove these parasites from the skin of the children, provided antibiotic creams and even shoe, because those people they can't afford shoes, so they walk around barefoot, and these shoes also prevent this disease to be spread. So now you may be wondering, does it really work? So we have so many plants that the animals use. And I'd like to give you some insights on what happens in the lab. Of course, lots of samples are coming in, so we have to do a lot of extraction management, and with standardized method, we have method for upscaling, so technically we could even work in a semi-industrial or technical scale. And all these extracts are then at some point As a result of this extraction word added to our crude extract library, we created that where I started doing this during my PhD, and now we're being a little bit more and more senior, we continue this work. And it currently encompasses more than 700 unique extracts from more than 200 plant, insect, and mushroom species. You may know that 2 out of 5 plant species currently face extinction, so archiving this almost never-ending chemical diversity or diversity of chemical substances that nature produces for future generation will be vital also for the discovery of novel drugs in the future. The battery is quite low here, I always have to turn it on and off. So let's talk about some results of our work. For example, this berry here alone, it's a plant or berry known locally, umuhangwa, which is occasionally eaten by the mountain gorillas when visibly sick, and also by a bird here, that's what we saw. And we prepared an extract, which you can see here, I think a nice extract, a beautiful extract in terms of color. And through our in vitro studies, we have found that this particular extract is highly effective in killing clinical isolates of life-threatening, multi-drug resistant bacterial pathogens, those or multiple bugs where our conventional antibiotic therapies already become useless. But there's even more to it. It's also anti-inflammatory, and it was effective against disease-causing roundworms. All in vitro assays, though. So we haven't gone into any animal models or clinical trials, of course, in this case. What about Mbiribiri? Remember that plant that Tom ate just before his death. And this is unpublished data, so we hope to publish it soon. We found that this particular plant, this is the moment we collected it, possesses pain-relieving properties similar to those of paracetamol or acetaminophen, ibuprofen, aspirin, and diclofenac. So it's the same mechanism of action. And our lab results then also support the claims of the local traditional healers that we interviewed when we asked them, why do you think that this plant is medicinal? How do you use it? And they said, we use it to treat fever in young children. And so likely, this is maybe a little bit unprofessional to say, but I think it's also in theory correct. Tom treated the symptoms, the pain, the fever, but not the cause, not what caused the sepsis, the bacteria. Let's talk about 2 studies towards the end of my presentation that we've published. On behavior in Budongo rainforest, which is in western Uganda. So we observed that there's chimpanzees that ate 13 different types of barks from trees. So this eating them increased in seasons of respiratory infections, and they single individuals traveled long to access these barks. They prioritized them over meat or other high protein or high valuable resources. And basically there you can see some of the evidence. They stripped it off the trunk. So we have all of this, so it's evidence that certain barks may be medicinal. And this is the work of Elodie Freeman, Dr. Elodie Freeman, who's also an AC50, and it was part of her PhD thesis. Then she's a postdoc in my group right now. Then Dinda took over. This is a master research. She was a master student in my lab. And we published then her work after Elodie published her part on the field work. And we found that all of these trees have certain bioactivity, and some of them really strong antibacterial, anti-inflammatory properties against multi-drug resistant bacteria. And this was a huge study because more than 200 media agencies, TV, radio, print and online media were interviewing us and reporting about it. BBC did a piece. I even put I hope I pronounced this right, APACE, they did an interview, in English though, with me of course. And the Washington Post, Science wrote a piece, so it's really, zoopharmacognosy was all out there in the world. People were reporting about a niche field that's normally not really relevant to many people. Another focus of my work is the fight against, I mentioned that antibiotic resistance, but also the search for new strategies that exert less evolutionary pressure, not like antibiotics. So basically, that less evolutionary pressure in conventional therapies, thus leading theoretically to less resistances. And one of the strategies we are looking at in the lab is the quorum quenching, or the inhibition of the quorum sensing. Quorum sensing is the ability of bacteria to communicate with one another, to join forces, to produce biofilms, to produce and exert toxins that may be the cause for death in humans or for losing joints and so on. And we've published 3 studies on 3 plants from Uganda that are used by humans for treatment of wounds and for bacterial infection, or what I would regard bacterial infection. And we found that none of these plants is actually killing the bacteria or harming the bacteria. They just grow and they live and they thrive. But what they do is they can disarm the bacteria. They can take away their ability to communicate and making them basically a tiger without fangs and claws. In German we would say ein zahnloser Tiger. And this is really, really cool, I think, because it's a new strategy that may work. And at the same time, does a plant necessarily need to kill or harm the pathogen in order to be effective and cure the disease? I would say not necessarily. And we can also argue that this can be put into like bandages and so on. So we're looking at that. And in terms of application of the research, we also, this is a grant application that we just submitted. It's a major grant, lots of funding with a startup called Pangea Bio, where we want to use big data and basically build one of the biggest databases of plant chemistry linked to human medicine, human biology, with millions of data points. And we're also using AI. Alicia will be talking about AI in a bit. And we are also trying to do that. I think all the researchers are applying AI, but in this case we have 3 different modules. One is basically mapping relationships between species, humans, human uses, compounds and so on. And then we have the computation of metabolomics and compound activity profiling, where we try to accelerate drug discovery rates or the process of drug discovery and increase the rates, the success rates, because that's traditionally a problem with natural product drug discovery. So getting scalable product pipelines and then having them ready for further development. The special thing about this is, we have an extract. An extract is complex. There's more than 1,000 or around 1,000 different compounds in there. How do you know which compound is actually the one that's causing this effect? And getting this out of it's a whole PhD thesis. So it takes years. Isolating this compound. And in the end, you end up with a compound that's not really bioavailable. It's not really active anymore. Synergistic effects were the cause of multiple compounds, so you're losing the activity. Or it's maybe just toxic. Or it's a known compound that's already discovered and it's not really useful for any reason. So what we're trying here, my whole library, basically we create fingerprints, chemical fingerprints through mass spectra, and then we dereplicate the known and predict novel structures in the compounds in there, before ever taking the extract to an assay, an experiment and looking at the activity. And then we already know what we want to do and what we are looking for, and then we do the validation in the lab. My last 2 slides, current projects. I just want to mention that we have a paper with Alicia as well. She's also co-author here, where we look at Kenyan elephants, and I hope that we can sit down tomorrow and finalize and just write the abstract, and then we can submit it for publication. And there's a project that we started earlier this year. It's also where Candice is working. It's on exploring the spiritual, ritual, and pharmacological facets of psychoactive, psychedelic materials used by humans and animals in Tanzania and Uganda. And I'd just like to highlight there's lots of collaborators, but one of my favorite collaborator, I think, is Jane Goodall, who's childhood hero, and now working with her and mentoring, receiving mentorship from her. And my last slide coming up. Yeah. So yeah, feel free to follow me on social media, and thank you for being here. I'm going to play this.
Okay, so here I am with Fabien, and I want to say hello to all of you, his students. I've been hearing all about the work that you're doing. It's really, really important, and I hope that some of you will follow in Fabien's footsteps and really help to change the world, to change the future, to save lives, and to find out more about the chimpanzees.
Okay, thank you very much. I'd like to acknowledge my group, of course. And, yeah.